Mubin and I have actually met before. We're doing a very similar thing in our jobs where we're taking science and working in hospitals and trying to apply machine learning and AI, or maybe not so much, as we'll hear, to different technologies. And so Mubin, I would like to hand over to you to talk about all things at Navi.
Thanks. I'm so tempted to talk about mechanical keyboards instead of this after watching Sarah's presentation. You know, that's a hobby I have, but try to stick to this. My name is Mubin. I'm one of the co-founders and CTO at a early stage, not so early stage, but a startup company from Melbourne called Navi Medical Technologies. At Navi, our vision is to create brighter healthcare features for critically ill children. Basically, a fancy way of saying we build medical devices to improve care for sick children. Today, I'd like to talk about the journey that we took as a bunch of founders and students, and coming out of uni and creating this startup and reaching the point that we are at now. I'd like to talk about the problem we're solving, the solution that we're building, and also some of the challenges that we have faced. It may not be as exciting as Sarah's presentation, but I'll try my best.
It's more useful, much more meaningful.
I know. Should I just control myself?
Yeah, if you just press up and down from here.
So a quick, next to the speaker.
So apologies guys, I'm going to have to go because I have a Christmas light show turning on in 35 minutes, and I actually can't be there when it goes on, which is the story of my December and why you shouldn't do this.
Yeah. All right.
I'll keep going, nothing. It's all right, they're just going to grab me laptop now.
No worries. Yeah, I'll just quickly go through my career summary. So I'm a biomedical engineer. I did my master's degree from University of Melbourne in 2015. Graduated in 2017 and worked as a research engineer in heart research at Murdoch Childrens Research Institute, which is where I work a lot of AI and image processing stuff. Murdoch Childrens Research Institute is located within the Royal Children's Hospital, and concurrently, while I was doing my degree and working at the RCH, or Murdoch Children's Research Institute, I co-founded Navi, and I'm currently the CTO. I'd like to talk about the journey, like why I was doing it, then how I was doing it while I was studying. But it's an interesting journey, so let me move to the next slide. Okay, so this is the Navi founding team. We actually have 6 co-founders, very unusual, or I don't know, it might be usual, but back then when we started a company, a lot of different people in the domain said that we have a relatively big company and it's going to be very hard for you to manage a lot of different things. It's quite risky. But here we are, 7-8 years later, we're still ticking, and we have done a lot of things since then. But interestingly, when I was in uni, in 2016, the University of Melbourne introduced a course called Biodesign Innovation. And the purpose of that course was to bring together students from the School of Engineering that were doing biomedical engineering and students from Melbourne Business Schools who were doing part-time MBAs. And the purpose was for them to form a cohort where a bunch of teams would come out. The teams would have advice from a bunch of different clinicians around Melbourne, clinicians from paediatrics, cardiology, respiratory. There was a lot of different clinicians that were part of that course. So the purpose of the course was pretty simple. You go out, you talk to more clinicians, nurses, doctors, and figure out the problems and unmet needs in the medical domain. They call it the biodesign approach. And once you have found a problem that you're really passionate about or you want to really solve, you come up with a solution, and then you present a business case as part of the course. Now, for us, once we graduated from that course and eventually our degrees, we took a break, came back together, and actually decided to form a company out of it, mainly because the team that we had built as part of the course, we really enjoyed working together, and also the need that we were solving, the unmet need that was in the neonatology and pediatric domain, was so urgent and was so needed by different people around the world that we were like, okay, this is something that we really got to work on. So we had some motivation from there. In about 2018, we got serious, started joining a lot of accelerator programs, and I think I was telling, was talking about it, but we were at Stone and Chalk in 2019 as part of one of the accelerator programs here called Actuator. Stone and Chalk used to be in the good shape then. But yeah, anyways, 2018 is when we all went full-time, or 3 of us went full-time, and the rest of the founders were part-time. Since then, we have done 3 clinical studies. We have studied about 150 patients at the Royal Women's Hospital in the neonatal intensive care unit. We have done more than 200 clinician interviews across Australia and US, different hospitals, children's, neonatal, 30 plus product demonstration, and have developed multiple iterations. I'll talk about all that in a bit, but we have raised about more than $10 million so far, and we have 12 people in the team now. So I think from considering that we started as students from classroom and getting to this point, I think I feel really good that we have been able to reach this point, and we still have the same people that were involved from the very start. But now to the actual bit. So what are we trying to do? So the problem that we're trying to solve has to do with something called a central venous catheter. Central venous catheters are basically thin flexible tubes that are inserted either via the arms or via the legs, any of the limbs, to get access to the heart. And the purpose is to deliver medications, nutrients, and fluids directly to the heart, because there's so much turbulence in there that you can easily, that the medication can easily get transmitted throughout the body and at a pretty quicker time frame compared to if you're getting it through nasogastric tubes or feeding it directly through the mouth. So that's the purpose of the central venous tube. But problem with placing central venous catheters is that it's done under the skin, so it's a blind procedure. So the way it works is that the clinicians would do some sort of guesswork to figure out how much they're going to be inserting the catheter in, and when they think they're in the right spot, based on haptic feedback, some of the blood returns, and a few other things that the clinicians do, they'll call for an X-ray. When the X-ray comes in, it can take about an hour depending on the time of the day. If the X-ray says it's not in the right spot, which could happen about 40-50% of the time, we'll have to sometimes adjust the line or completely remove it and introduce a new line, and, redo the procedure, and it's just back to square one. So there's a lot of frustration involved with this procedure. There's also, because you're doing multiple X-rays to re-verify if it's in the right spot, there's a lot of ionizing radiations that you're providing, putting on to the patient. And if you think about newborn babies, premature babies specifically, who require these lines in emergency care, and they mostly get it through the umbilicus. It's quite important because the first hour is a golden hour where they need urgent care, and because of this shortcoming, a lot of the time that care is delayed. It's pretty bad for the baby because it can easily get fatal. Okay, so that's one problem. The other problem is that even when the line is in the right spot, it has been secured, and now the X-ray says it's in the right spot, about 28 to 75% of the time, in just the first week, the line migrates into a suboptimal location. Now this is even worse because by this point, they have started delivering drugs and medications which are toxic, and they're only intended for that particular area of the body. So if, let's say, the tip goes too high, like it gets gets into the heart, it can actually inflate the heart's outer layer and cause something called cardiac tamponade, which can require serious intervention and could lead to death. There are a lot of other complications that could arise because of this migration problem. The issue is that there is no current way of knowing if the tip is in the right spot, and the recommended way of checking for it is you do an X-ray every day, which is also dangerous because you're blasting a baby with radiations pretty much every day, and it's not good for them. It's ionizing radiations. So this is where we come in. The system we're creating is called the Neonav ECG tip location system. We are trying to solve the problem of the lack of real-time feedback, and the way we do it is through a technique called intravascular ECG. How many of you know what an ECG is here? It's all right, a lot of people. ECG is short for electrocardiogram, and it's the electrical activity associated with the heart. So when your heart pumps, it's basically an electromechanical chamber. Brain is sending a bunch of electrical signals, and you can actually pick those signals up if you put certain electrodes in certain positions on the patient's body, and you can use that to diagnose a bunch of different things. You can actually use that to diagnose heart blocks and heart attacks, but that's not what we're doing here. What we are doing is that we are converting one of the surface electrodes, or we're converting the catheter into one of the surface electrodes. So what happens is that as the catheter moves closer to the heart, we get different characteristics, and we pick that up and send it back to our machine, and we have a smart algorithm there that recognizes the changes, and based on that, it's able to say whether the tip is in the right spot or incorrect spot, and that's how you get the real-time feedback. Any questions so far? Cool.
And I mean, do you actually get like a visual imagery, or you don't get a visual imagery?
No, at the moment, so we talked to a bunch of users, and they just wanted 3 colors, red, blue, red and green, basically just 2 colors. And we still display waveforms if they want to make their own judgment. But yeah, just red and green is what they're after, and not, where it is in the body. Because there are basically 3 things that they want to know: whether they're in the right spot, whether they're too deep into the heart, or whether they're too short or in an unintended vessel. Once they know that, it's so much easier for them to just, move the catheter in the right spot. The way we pick up the signals to the catheter, I think that's a very interesting part, because we don't put anything on the catheter. The catheter is off the shelf. The device can work with any central venous catheter for any patient, not just newborns, pediatrics, even adults. What we do is we attach something called a catheter connector, which we renamed it to the Neonav ECG adapter. Don't ask me why, it's just the connector makes it so much harder for the documentation, because sometimes you'll be saying the connector connects to the cable. It just becomes a tongue twister. But the Neonav ECG adapter, it's a very simple piece of equipment. It's a dumb equipment. What it does is that it attaches to the proximal end of the catheter, and clinicians normally flush saline through the catheter. So it makes the catheter an electrically conductive probe, which as it moves through the heart or through the vessel, you get different signals. But this adapter converts the saline column or the saline signal that we're getting through the tip of the catheter into electrical signal. So there's basically a tipping electrode inside that converts the ion current into an electric current. It's just a piece of metal, it's nothing fancy. And that goes back to an acquisition unit that sits next to the patient, and that does some sort of processing, but then it then gets passed to the console where we have the software. Does that make it clear?
How does it decide what is the right position? Because it's a single dimensional reading, isn't it?
Yes, so it's coming from the catheter, but it's also comparing the signal with an electrode that's placed on the body. So there's still a written pathway. There are 3 electrodes placed on the body. We still get the normal surface ECG, but then we also use one of the electrodes to compare the signal against that. Now the way it decides, it's based on the algorithm. And that's what I want to talk about is, we have developed an algorithm based on a lot of clinical data that we label with X-rays or X-ray interpreted applications, and then we were able to use that to, run the system. We have a remote as well, as requested by the users. We've done a lot of user interviews that were like, it was awesome if I was able to place the line, but at the same time control the console. So I put in a 3-button remote that has a sterile cover on it, and you can use it in a sterile field because it's a sterile procedure. You can't put the console in the bed itself, so it's much easier if you have a remote. But now I'd like to talk about some of the challenges. Okay, so in theory, it sounds very straightforward, right? Like you have an algorithm which is basically looking at a lookup table of a lot of ECG segments that are labeled with different tip locations and making a decision based on, which one matches based on what you're getting in live, right? But getting that data has been the bane of my existence. I think I've lost a lot of hair because of that. A lot of my hair has been gray. You can't see them, but they're there. But I would have had a fuller head if it wasn't for that. But there were 3 to 4 challenges that really bothered us and really, slowed down our journey. The first one was the hospital approval hurdles. Now, when you're trying to collect data from a hospital, or when you're trying to do anything in a hospital, you need to go through multiple committees from the submission all the way to recruitment. A lot of different departments have to look at your application and approve it before you can actually use the data for anything at all. So in our case, we worked at the Royal Women's Hospital, and there were a lot of these different departments that our application went through. So you had the research governance, which looks at the research side of the protocol, the scientific validity. Do we have any agreements with the hospital, etc., etc. That took some time. We had the HRAC, which is the Hospital Research and Ethics Committee. Just looks at the ethical implication of the study. occupational hazard, obviously, from introducing anything into a clinical setting, there are bound to be occupational hazards, and how do you, mitigate those? You have to talk about that. Infection control, it's a sterile procedure. There's always risk of infection. How do you prevent that? You have to notify the TGA, which is the regulatory body that regulates medical devices in Australia. Clinical database entry and receipt, you have to file a, it's part of it's mandatory now for you to file your clinical trial into a database. And then you have to wait for them to give you a receipt, and then you give it back to the HREC, and then you can run the study. So convoluted, but the final one, which I would like to call the final boss, was the BME approval, which we underestimated a lot. So BME stands for biomedical engineering. So there's a biomedical engineering, and I'm a biomedical engineer, and I'm so sad how long it took. But biomedical engineering departments are the final sort of gatekeepers before you can actually start recruiting, because they make sure that the device is safe themselves before you can use that in a study. So this is how one of our studies timeline looked like. So you can see that the HREG and RTO reviews, they were relatively quicker, about 2 to 3 months, and the TGA database entry, which is occupational hazard and infection control, were like 1-2 months extra. But then the BME review really took the toll. Like that was easily 7 to 8 months. And okay, for a big company, 7 to 8 months might not be nothing. They could be focusing on the product. But for a startup like ourselves, who's running on burn rate, that's pretty massive. 7 to 8 months multiplied by, I don't know, if you use 50K, that's still a lot of money that you're just bleeding, considering you have limited money. But that was how it was. With some of these problems, what I wanted to do was talk about the problems and talk about the solutions as well that we have been able to implement. We haven't been able to find a really good solution that would work with the approval hurdles. You just can't go through red tape unless someone really close in one of these departments that can help you speed it up. In our case, we have put some agreements with the Royal Women's Hospital to ensure that in the next study that we're trying to get approved, the BME approvals are faster, but I'm not holding my breath for that. Okay, so navigating data capture in ICU. So once we started patient recruitment, one of the biggest challenges for us was, so we were basically collecting data from umbilical venous lines, which are placed in premature babies or newborn babies. So babies, as they're born, if they're premature or if they need emergency care, these are the babies that are eligible for these lines. Now the issue is that these babies are born all the time, right? These procedures are done like 300 procedures done in a year, and most of the time, these procedures are done at night. So there's no planning. Sorry, there is planning, but there's a very small window for you to know that the procedure is happening and you have to get there. It's mostly at night. So that made it so difficult for us to get to the actual procedure and collect the data, because what would happen is that, if you would get a call, we'd only have like a 20-minute window to get there before they start the procedure, because it's an emergency procedure. They can't delay it because of a study. But a lot of the times, the nurses just wouldn't call us. They would forget about it. Like there's a big sticker on the bed saying, please call us, the Neonav study. If you see there's an umbilical venous line getting in. So that was very frustrating. Fortunately, some of us who don't have lives live nearby, and the latest data capture that I did was at like 1 a.m. when when twins were born. Was the awesome night of my life, because the data we got was super awesome. But I was there for 2 hours, 2.5 hours, and just there, just collecting data, enjoying my life, I guess. I guess you got to do that if you're a founder. We have nurses now, but initially we didn't have any nurses. We were like 6 people, and 2 of us were just going in to collect the data. I went in because I lived close by. There are a few things that we did to make sure we can collect more data. One of the things was that, so this is the line insertion procedure that I'm talking about, when the line is actually being introduced into the patient, right? So these are very time-sensitive procedures, and they have to be done in an emergency setting. But what we also did was we wrote in the protocol that as the line has been placed and it has been sutured, we'll come in every day and we can come in at any time or connect our device and collect the data, which is awesome because we were getting more ECG data. And lastly, the thing that we did was when the line was being removed, because the line stays, it has delivered its care, and now it's time for removing. We said, okay, that's the time we'll come in. Now the good thing about line removals, where they weren't emergency removals, they can be done within a relatively bigger time window, and the nurses were more responsive because most of the time it was during the day. So we went in for the line removal. The thing was that with line insertion, we needed data at every centimeter of the catheter depth. But with the removal, you can still get that because it's in the right spot and you're just pulling back and you're stopping. And because it wasn't done by a doctor, it was less stressful, and I'm not saying doctors are more stressful, but this is a stressful procedure. It was done by a nurse, and because it was less stressful, it was easy to ask the nurse to stop at certain points. So data quality was really high. So that's the data capturing stuff. The next one was, when we got the data, what problems we had then. One of the biggest problems we had was data labeling disparities. So as you saw that we had multiple ways of collecting the data: insertion, adjustment, removal, and spot checks. Adjustment is basically when they do an X-ray after the insertion, they figure out that the line is not in the right spot and they have to adjust it. That's another way for us to collect the data. The issue was that we were only able to label 24% of our data with X-rays, so the ground truth. So for machine learning nerds, you would know that you have a data set and you need to label it with ground truth. We didn't have ground truth for everything. Only 24% of our data set had the ground truth, and that was because we couldn't add X-rays as part of the study because it's ionizing radiation. So we could only rely on the X-rays that were done clinically. For insertions, as you can see, most of the X-rays were done clinically because they do it to confirm the tip location. But for spot checks and removals, you basically had to get lucky. I mean, I shouldn't say that because they would only do an X-ray if they found there was a problem with the baby and they had to look at their chest. And we would get lucky in that. I shouldn't say that. But as you can see, there's a big disparity. We were only able to get certain amount of data, and that's a big issue. People who are in data science would know that when you have a missing label, there are so many different ways you can label them. But what do you do when you have you have clinical data that's not out there, and it's sort of a new thing? What we were able to do, a few things. One was that we were able to, we don't want to use the word interpolate, but basically predict what the locations would be for a lot of the data, and that was based on, so for example, if an insertion was done or if a removal was done, we would have the whole image, and we would know the centimeter readings for that, but we also took different ECG recordings at different centimeter readings as the line was going in. So we can look at the X-ray screen and we can correlate the anatomy with the ECG data. So it wasn't one-to-one, but it was indirect, but you could still be relatively confident in saying that it's very likely that that particular anatomical part in the X-ray belongs to an ECG segment that we took at 5 centimeters, 4 centimeters. So that was one of the ways we filled in the gaps. The other way was we did some lit reviews and found people have collected similar data sets, and we used that information to label our data set. So we basically had two-tiered data sets. One high tier or tier 1 data set was where we had actual labels, and tier 2 was the one where we did lit reviews and all the other stuff. So basically we had to figure out which one to use for development and testing. One of the things that we're doing now for our next study, which has already started, is we have introduced bedside ultrasound. So one of the other ways you can actually get the ground truth is by doing point-of-care ultrasound, and point-of-care ultrasound doesn't require someone to come in. It doesn't have ionizing radiations, so it's not unsafe for the patient. It's just a few extra minutes after the procedure where you can put a probe and figure out where the line is. And we have been using that to increase our data set, and that data set has been really good, and we've been able to use that for training rather than training. We have been able to use that for testing rather than training. The last problem, pivoting from machine learning to traditional methods. So we started with machine learning approach, training the data and testing it. But there was a big issue from a regulatory point of view, because when we presented our data to the FDA as part of something called a pre-submission, which is not a submission, but a meeting that you can do before submitting your device, FDA had concerns about data integrity. They still haven't put in a final guidance around how they regulate machine learning applications unless something has changed in the past few months. Black box is a big black box for them. Because they don't know what's happening inside, they're really hesitant in approving devices like that. And what they ask for is that you have a really strong data pipeline. For our particular application, this meant that you don't just collect data from one hospital. You go to 10 different hospitals and collect as much data as you can. You can't rely on just 100 data, even though it's 100 data points, even though it's statistically significant. You still go out, you collect 500 more data points from different hospitals. It took us 1 year to get approvals and 1 year to actually collect like just 50 patients. Imagine if you were to go to different hospitals. So that was one of the reasons we had to pivot. But the other one was that they wanted a validated prototype to be put in the clinical setting to collect the data. When I say validated prototype, this means that basically the final device that you're going to be putting into the market has to be the one that collects the data. Now, it was quite impossible for us to do that because we were using the data to develop the device. So we had no way of using that and sort of retrospectively change the data. There are ways to work around these. We now know that. But because we had already gone through the clinical studies and we had already collected data and we had met FDA afterwards, this was a big shock for us. So we had to pivot and we went for traditional statistical methods. We weren't able to get the same level of accuracy, but it was still pretty good and pretty close to what the clinicians wanted. And just dealt with a bunch of thresholds that we derived statistically. Put in a very obscure meme there, but I hope you get the idea. Lastly, just wanted to show that with the 3 clinical studies that we have done so far and 200 user interviews, we have gone through 3+ product iterations, and these are the major ones. So initially, when we had just graduated from the university and we were in year 1, we had this lunchbox prototype where you can see that there's still some leads that are visible. It was tested, but it wasn't good enough for a clinical setting. So we did a lot of animal studies and benchtop studies with that. The second one is our clinical prototype, which we developed based on the learning from here. We went out of our way to do a lot of benchtop testing to make sure that it's safe for use in humans, and that is the one that we used to collect like 50, 100 patients, more than 150 patients. And the last one is a current prototype. It's the design control prototype. It has all the right documentation that you would need for an FDA submission. It's basically a derivative of this, but with more bells and whistles. It looks pretty. The usability is quite enhanced. The signals look way more, user-friendly. But that's where we are at, and I thought I would show you, the whole journey, and this is where we have landed. In the next few months, we're trying to actually submit this and the documentation associated with it to the FDA. So let's see how that goes. But yes, that's all from me. Any questions?
So with the pivot from ML to statistical models, how are you finding the accuracy? And second, how much faster is it for your use case?
Faster in terms of when you're getting an output?
Yeah, I don't know if inference speed was necessarily an issue, but I mean, presumably it's less resource.
Definitely.
Yeah.
Yeah, so, I mean, because our data set isn't as big, machine learning versus traditional methods didn't really pose any challenges around speed of development. I think your original question was the accuracy difference. I think it was about a 5% hit that we got, but there's still ways that we can improve if we're collecting more data and we're seeing ways. But yeah, it wasn't that bad. I mean, I was worried it's going to be that bad, but it wasn't, which I'm glad.
With the regulatory approvals that you have to seek, I assume that you have to combine a lot of documents and do you have an external support in doing that, or was it all you are doing that?
No. So we're just 12 people. Like, we're quite lean, and we only have like 3 engineers in the team. I do all of the software development, and I had to get trained into 6 to 3 or 4. But a lot of our other engineering work, a lot of our regulatory work, quality work, clinical work is outsourced to different firms. Found that the Australian firms are okay, but the American firms are way better because they've done it so often. With Australian firms, or with any firm in general, when you're working with them, I think you got to know the risks and you got to know how to manage them.
And one more question. So I see that X-ray is going to, X-ray technology is going to be a key part in validating your data and being able to use that more efficiently. So have you looked into new advancements in X-ray technology that can help you?
Like machine learning and all that,
Like
that.
soft X-rays which are not ionizing as much as your hard X-rays, which could help you validate or which could help you label your data in fact. Something like that.
So the type of X-ray technology being used is really a function of the hospitals that you go to. You can't go beyond what they already use. You can try to introduce it in, but that's like another year of a process.
So maybe just extending that, right? You're saying that you can measure at every centimeter, maybe like the feedback, latency. So is that what you're talking about, the limitation on the hospital room?
So when I said centimeter measurements, I mean that as the catheter goes in,
Yeah.
there are actually markings on the catheter. So you can see the marking on the outside and you'd be like, okay, it's 5 centimeters in, 4 centimeters in. So the idea was that at every centimeter, you collect an ECG segment, 5 seconds, 10 seconds. And then on the X-ray, even though you're only looking at the tip location of the final point or the final insertion depth, but you can sort of extrapolate the remaining ones just by doing centimeter measurements on the X-ray itself to know, because you're still taking the ECG readings, right?
Yeah.
And you still have the X-ray, so you can use the image in more ways than possible, I guess.
And my question is around like the unlabeled data sets, because I find that really fascinating. You do like smoke techniques and all that stuff. So did you go into like, because you're saying you use proxies to kind of identify signals for that. So did you look into like PCA and kind of find other signals that could be meaningful that you didn't think you can expect to be good?
So one of the AI technique that we kept in, that we didn't get rid of when the FDA told us, and there's no reason to get rid of it, is some of the principal component analysis and some of the feature analysis that you can do using machine learning techniques. That actually helped us figure out, well, because it's an ECG, you have P waves, QRS waves, like you get a very nice waveform and there are different characteristics of it. You have amplitudes, you have durations, you have shapes, so many things that you can take out of it and you can try to characterize them based on, like you can use different techniques to figure out which features are sort of the best ones. So yeah, we did use some techniques there.
Shall we?
Well, I just want to say, if you're submitting in the next month, as I'm at the exact same stage, it's no mean feat doing 62304 regulatory software development practices, so I feel you. Thank you so much for your talk. It's really interesting hearing about all the techniques that you've applied. I did have one question. So you use PCA to do the feature extraction, and then you use those features to then do more traditional modelling.
Yes, not exactly PCA, but that was one of the techniques that helped me focus on the right. Because the one good thing about this was that I was actually present in the clinical settings. So when I would go into the clinical settings, I can actually see a lot of the things that you weren't able to record because of constraints on data recording. Like I would see exactly at each second how the ECG would change. So I had acquired some, how do you say it, like knowledge that wasn't documented. So I used some of that as well to figure out the features. But then there were obvious features that you can just do qualitative analysis on them and be like, okay, the PO is changing differently, the QRS is changing differently, and you can use that. But the PCAs and some of the machine learning techniques really help me focus on the right.